Hello and welcome back to the Sleepyhead Geographer. I'm so glad you've joined us tonight for another gentle journey into our world's most fascinating mysteries. Before we begin, let's take a moment together to settle in. Find your most comfortable position, perhaps pulling that soft blanket a little closer. Let your shoulders drop, release the weight of today's worries, and allow your breathing to slow and deepen. Tonight, we're leaving behind the rush of daily life and venturing somewhere truly extraordinary. If you're enjoying these sleep stories, a gentle like, share, and subscribe helps others find this calm space too. Now imagine discovering something so startling, so impossibly vibrant against this frozen canvas, that it would capture the imagination of explorers and scientists for over a century. We're going to explore the enigmatic Blood Falls, where crimson water cascades down pristine ice like something from another world. This remarkable phenomenon has puzzled researchers, challenged our understanding of life itself, and revealed secrets hidden beneath the ice for millions of years. So let's embark on this peaceful journey together, following the story of one of Earth's most beautiful and mysterious natural wonders. Let us drift back through time to the year 1911, when the world was a much larger place, and Antarctica remained one of Earth's greatest mysteries. The wind was sharp that austral summer day as British geologist Thomas Griffith Taylor made his way across the rocky terrain of what would later bear his name, the Taylor Valley. The expedition was part of Captain Robert Falcon Scott's Terra Nova mission, and every step into this frozen wilderness brought new discoveries that would reshape humanity's understanding of our planet's most remote continent. Taylor moved carefully across the valley floor, his boots crunching on the gravel and ice that had been sculpted by millions of years of glacial action. The McMurdo dry valleys stretched around him like an alien landscape, one of the most extreme deserts on Earth, where the katabatic winds sweep down from the polar plateau with such tremendous force that they scour away snow and ice, leaving behind bare rock and ancient soil that has remained undisturbed for millennia. The silence here was profound and almost overwhelming, broken only by the whisper of wind across stone and the distant groaning of glacial ice as it slowly moved through the valley system. As Taylor approached the terminus of the glacier that wound down through the valley like a frozen river, something extraordinary caught his trained geological eye. Against the brilliant white ice, a stain of deep red cascaded down like spilled paint from an artist's palette. The sight must have been breathtaking and bewildering, a waterfall of what appeared to be blood discharging from beneath the glacier. In this land of endless white and pale blue ice, here was a color so vivid, so alive, that it seemed to pulse against the sterile landscape like a beating heart in a frozen chest. Taylor approached the phenomenon with the careful eye of a trained geologist, but surely his heart must have quickened as he witnessed something that challenged everything he understood about Antarctic environments. The liquid flowed clear from its source beneath the ice, then transformed before his very eyes, quickly turning crimson as it met the Antarctic air. The contrast was startling, like watching invisible ink reveal its secrets. The transparent water blossomed into shades of rust and scarlet as it tumbled down the glacier's face, creating abstract patterns that shifted and changed with each moment of flow. The early explorers of Antarctica were no strangers to the unexpected and the sublime. They had witnessed the ethereal dance of the aurora australis, painting the polar sky in sheets of green and blue light. They had heard the thunderous carving of icebergs as massive chunks of ice broke away from glacial fronts with sounds like artillery fire. They had felt the raw power of katabatic winds that could knock a person flat and create whiteout conditions in minutes. But this crimson waterfall was different. It seemed almost biological, almost live in a land where life appeared not just scarce, but utterly impossible. The Antarctica pioneers first attributed the red color to red algae, a reasonable hypothesis for the time given their understanding of extremophile organisms. After all, they knew that certain algae could thrive in harsh conditions and produce vivid pigmentation as protection against intense radiation and temperature extremes. The idea that microscopic plant life might be responsible for this dramatic coloration seemed logical, even comforting. It suggested that even in this most desolate of places, life might find a way to announce its presence in spectacular fashion, creating beauty where survival itself seemed miraculous. Word of Taylor's discovery spread through the scientific community with the measured pace of early 20th century communication, carried by letters and telegraph messages that took months to reach the universities and geological societies of Europe and America. Letters and reports made their way back to institutions like the Royal Geographical Society and the American Geological Society, carrying descriptions of this remarkable phenomenon that captured imaginations and sparked heated debates in lecture halls and scientific journals far from the Antarctic cold. The phenomenon would come to be known as blood falls, a name that perfectly captured both its visual drama and its mysterious nature. For decades following the initial discovery, the red algae theory persisted with remarkable tenacity in scientific circles. It made sense within the framework of what scientists understood about extremophile organisms, life forms that thrived in conditions that would kill most other living things. Researchers knew that algae could survive in hot springs, where temperatures reached near boiling, in highly acidic environments that would dissolve metal, and in places with extreme temperature fluctuations that cycled from freezing to thawing on daily or seasonal basis. Why not in the harsh environment of an Antarctic glacier, where the challenges were different but no less extreme? The location itself added layers of mystery and intrigue to the phenomenon. Taylor Valley sits within the McMurdo Dry Valleys, a region so extreme that NASA has used it as an analog for Martian conditions in training astronauts and testing equipment designed for planetary exploration. Here, temperatures can plummet to -50 degrees Celsius during the polar winter, creating conditions where exposed skin can freeze in minutes. The air is incredibly dry, with humidity levels lower than some of Earth's hottest deserts, and the UV radiation is intense due to the thin atmosphere and constant reflection off snow and ice surfaces. It's a place where mummified seals from centuries past lie preserved by the dry, cold air, a natural museum of Antarctic life and death that speaks to the preservation power of extreme environments. As more expeditions ventured to Antarctica in the decades following Taylor's discovery, Blood Falls became a landmark, a waypoint in the vast white wilderness that helped explorers navigate the featureless landscape of the dry valleys. Explorers and scientists would make pilgrimages to see this crimson cascade, each bringing back their own observations and theories that added pieces to the growing puzzle. Some noted that the flow seemed intermittent, appearing more pronounced at certain times than others, suggesting complex dynamics beneath the ice. Others observed that the staining extended beyond just the waterfall itself, creating rusty patches on the glacier and surrounding rock that told of years of mineral-rich water flow. The mystery deepened with each careful observation and scientific expedition. The waterfall didn't behave like other Antarctic water features that researchers had studied. Most glacier melt in Antarctica is seasonal and relatively predictable, tied to the brief summer warming when temperatures rise just enough to create surface melting. But bloodfalls seemed to flow according to its own mysterious rhythm, sometimes appearing more vibrant and active, sometimes almost dormant for extended periods. This inconsistency puzzled researchers and added another layer to the enigma that would take decades to unravel. Photography from these early expeditions, though limited by the technology of the time, captured the stark beauty of the phenomenon in ways that words could hardly convey. Black and white images somehow made the contrast even more dramatic. The dark stain against the light ice created compositions that were both beautiful and unsettling, like abstract art painted by geological forces. These photographs would travel the world, appearing in National Geographic magazines, scientific publications, and Geographic Society presentations, spreading awareness of this Antarctic wonder to audiences who would never have the opportunity to witness it firsthand. The indigenous knowledge systems of the Antarctic continent were limited, as no native human populations had ever established permanent residence in this harsh environment. Unlike other mysterious geological features around the world, bloodfalls had no folklore or traditional explanations to guide early researchers. There were no creation myths, no ancestral stories, no traditional ecological knowledge to provide alternative frameworks for understanding. It was a purely scientific mystery, waiting for the tools and understanding of modern research to unlock its secrets through systematic observation, hypothesis testing, and technological innovation. As the 20th century progressed and scientific methods advanced with new instruments and analytical techniques, the simple algae explanation began to seem insufficient to explain all the observed characteristics. The color was too intense, too consistent, and too localized to be explained by simple plant life growing on or near the glacier surface. The chemistry seemed too complex, the behavior too distinctive. Researchers began to suspect that something more complex was at work beneath the ice, something that would require new tools, new thinking, and new approaches to understand fully. The bloodfalls had announced themselves to science with their stunning visual display, but they had also presented a challenge that would take nearly a century to fully comprehend. In this harsh land at the bottom of the world, where survival itself was an achievement worthy of celebration, nature had created something beautiful and mysterious. A crimson river flowing from the heart of an ancient glacier, carrying secrets that would revolutionize our understanding of life on Earth and expand our conception of where life might exist throughout the universe. To understand the crimson mystery of Blood Falls, we must first journey deep into geological time, back millions of years to when Antarctica was a very different place entirely. Let us settle gently into this exploration of ancient landscapes, feeling the weight of deep time resting softly on our imagination as we discover the remarkable foundation upon which this modern mystery stands. The story begins not with ice and cold, but with warmth, life, and the slow dance of continents across our planet's surface. The McMurdo Dry Valleys, where our bloodfalls reside, represent one of Earth's most extraordinary geological theaters, carved and shaped by forces operating on timescales that dwarf human experience. These valleys were sculpted not by the gentle persistence of rivers flowing over centuries, as we might find in temperate climates, but by the immense grinding power of glacial ice during multiple ice ages that advanced and retreated across the continent over millions of years. Picture these ancient glaciers as massive sculptors, their icy tools weighing billions of tons, slowly but relentlessly carving through solid bedrock grain by grain, creating the distinctive U-shaped valleys that characterize this region today. The bedrock beneath the Taylor Valley tells a story that spans hundreds of millions of years, written in layers of stone that preserve the memory of ancient environments. The foundation stones here are primarily composed of Beacon supergroup sandstones and Ferrar dolerite, rocks that were formed during the Devonian and Jurassic periods, when this land occupied a very different position on our planet's surface. Back then, roughly 400 to 180 million years ago, Antarctica was part of the massive supercontinent Gondwana, positioned much closer to the equator where the climate was temperate, even subtropical in some periods. During those ancient times, forests of seed ferns and early conifers grew where now only bare rock and ice exist in harsh desolation. Rivers meandered across landscapes where today we find only the whisper of katabatic winds carrying fine particles of ancient sediment. Shallow seas periodically flooded the region, depositing layers of marine sediments rich in organic matter from primitive marine life forms. These sediments would prove crucial millions of years later, providing the chemical foundation for the remarkable ecosystem that exists today beneath Taylor Glacier. As we drift deeper into this geological story, we encounter the crucial element that makes blood falls possible: the presence of these ancient marine sediments buried deep beneath layers of ice and rock. Millions of years ago, this region was periodically flooded by shallow, warm seas that teemed with early marine life forms. Trilobites scuttled across the seafloor, primitive fish swam in the water column, and countless microscopic organisms lived and died, their remains settling into the sediments that accumulated on the ocean bottom. These waters deposited layers upon layers of marine sediments, rich in organic matter and minerals, creating the foundation for what would eventually become one of Earth's most unique subglacial environments. The Taylor Glacier itself is a remarkable geological feature that extends approximately 54 kilometers from its origins high in the East Antarctic ice sheet, down through the valley system into Taylor Valley. This glacier represents millions of years of snow accumulation, compaction, and ice flow, carrying within its frozen structure the preserved record of Antarctica's climate history. The glacier is what scientists call a polythermal glacier, meaning that different parts of it exist at different temperatures. Some sections exist at the pressure melting point where ice can transition to liquid water, while other sections remain frozen solid at temperatures well below freezing. This thermal characteristic is crucial to understanding how blood falls functions, as it allows for the complex hydrology that feeds the mysterious red waterfall. Deep beneath the glacier lies a landscape that has been hidden from sunlight for an estimated 1.5 to 2 million years, sealed away like a time capsule preserving ancient environmental conditions. This subglacial terrain is not simply flat, ice-covered ground, but rather a complex topography of valleys, ridges, and depressions carved by ancient ice flows and geological processes that predate the current glacial configuration. The topography was shaped by earlier glacial advances and retreats, tectonic activity, and the gradual weathering of bedrock over geological time. Within this hidden world, a subglacial lake system became isolated from the rest of the world, creating conditions that would prove to be extraordinary for both chemistry and life. The formation of this subglacial environment was a gradual process that unfolded over geological timescales, requiring the precise alignment of multiple factors. As ice sheets advanced and retreated multiple times throughout Antarctica's complex climate history, the Taylor Valley region experienced alternating periods of ice coverage and exposure to surface conditions. During one particular glacial advance, possibly during the mid-Pleistocene period around 1.5 to 2 million years ago, the ice became thick enough and persistent enough to completely seal off a portion of the underlying terrain, trapping not just the physical landscape, but also the water, sediments, and even some of the atmosphere that lay beneath. This trapping process created a unique set of physical and chemical conditions unlike anywhere else on Earth's surface. The weight of the overlying glacier, eventually reaching hundreds of meters in thickness, compressed the trapped water and created immense pressure that fundamentally altered the physical properties of the liquid below. The isolation from the atmosphere prevented the normal exchange of gases and materials that would occur in surface water bodies, while the insulating effect of the thick ice above meant that the water beneath remained liquid even at temperatures well below the normal freezing point of pure water. The geology of the region contributed another crucial element to this subglacial system, an abundant source of iron and other minerals. The bedrock and ancient marine sediments beneath the glacier are extraordinarily rich in iron-bearing minerals, including pyrite, magnetite, and various iron sulfide compounds that formed under the reducing conditions of ancient marine environments. Over millions of years, the interaction between the trapped water and these iron-rich substrates would prove to be the key to understanding not only the crimson cascade visible at the surface, but also the complex chemistry and biology that thrives in this hidden world. The pressure dynamics within this subglacial system are fascinating to contemplate and crucial to understanding how the system maintains liquid water in such a cold environment. The glacier above acts like a massive cap on a pressure cooker, creating conditions where water can exist in liquid form at temperatures that would normally result in solid ice. The pressure from hundreds of meters of overlying ice effectively lowers the freezing point of water, while the high concentration of dissolved salts and minerals acts as a natural antifreeze, further depressing the freezing point. The result is liquid water at temperatures around -5 degrees Celsius, existing in a delicate balance of pressure, temperature, and chemistry that has persisted for millions of years. The hydrological connections within this subglacial system are remarkably complex and were long mysterious to researchers attempting to understand how water could move through such an environment. Water moves through an intricate network of channels, cavities, and fractures within and beneath the glacier, following pathways carved by pressure differentials, thermal dynamics, and the gradual deformation of ice under its own weight. Some of these pathways are temporary, opening and closing as the glacier shifts, settles, and responds to seasonal temperature changes. Others represent more permanent features carved by the persistent flow of subglacial water over thousands of years. The seasonal dynamics of the glacier also play an important role in the behavior of blood falls and the subglacial system that feeds it. During the brief Antarctic summer, when surface temperatures rise slightly and more meltwater is produced on the glacier's surface, some of this water can find its way down through cracks and crevasses, adding fresh water to the system and creating pressure changes that can affect circulation patterns within the subglacial lake. These seasonal variations help explain why the flow and intensity of blood falls varies throughout the year, sometimes appearing more dramatic and colorful, sometimes barely visible or completely dormant. The chemical environment created by this unique geological setting is unlike almost anywhere else on our planet. The combination of ancient organic matter from marine sediments, iron-rich bedrock, and the distinctive pressure and temperature conditions has created a subglacial brine that is both chemically complex and biologically active in ways that scientists are still working to understand. This brine contains dissolved minerals and organic compounds that have been concentrated and transformed over millions of years of isolation, creating a chemical soup that preserves elements from Earth's ancient past while supporting ongoing biological and chemical processes. Beneath the pristine surface of Taylor Glacier lies one of Earth's most remarkable hidden worlds, a subglacial lake that has existed in complete isolation for millions of years, maintaining its own unique ecosystem and chemistry in perpetual darkness. Let us descend gently into this secret realm, imagining ourselves floating through the darkness of this ancient water body that feeds the crimson mystery above, suspended in a liquid time capsule that preserves secrets from our planet's distant past. This subglacial lake system was trapped by the encroaching glacier at least 1.5 million years ago, possibly even earlier, creating what scientists describe as one of the most isolated ecosystems on our planet. Picture this hidden reservoir as a living time capsule, preserving not just water, but an entire chemical and biological environment from a time when Antarctica was beginning its transformation from a temperate continent into the frozen wasteland we know today. The water here carries within its molecular structure the chemical signatures of ancient seas, prehistoric life forms, and geological processes that operated under very different climatic conditions. The lake itself exists in a physical state that seems to defy our everyday understanding of how water behaves in nature. In our normal experience, water freezes at 0 degrees Celsius, transforming from liquid to solid ice in a process so fundamental that we rarely question it. But in this hidden world beneath Taylor Glacier, the laws of physics create a different reality, governed by pressure, chemistry, and isolation. The immense weight of the glacier above, hundreds of meters of ice pressing down with tremendous force equivalent to the pressure found deep in the ocean, creates pressure conditions that dramatically lower the freezing point of water through a process scientists call pressure point depression. Imagine floating in this ancient water, suspended in a darkness more complete than any cave system or deep ocean trench you might experience on Earth's surface. No sunlight has penetrated this realm for over 1 million years, creating conditions of absolute darkness that have persisted longer than most species of complex life have existed on our planet. The water around us would feel strange and thick, almost syrupy, due to its extraordinarily high salt content, nearly 3 times saltier than modern seawater. This extreme salinity results from millions of years of concentration, as the only water that leaves the system does, so through the tiny outlet that creates blood falls, while dissolved minerals continue to accumulate from the surrounding rock and sediment. The mineral-rich brine carries within its dissolved matrix the chemical remnants of ancient marine life, organic compounds that have been slowly breaking down and transforming in this sealed environment for geological ages. Carbon atoms that once formed part of primitive marine organisms now exist as dissolved organic compounds, providing chemical energy for the remarkable life forms that have adapted to this extreme environment. Nitrogen compounds, phosphorus, sulfur, and dozens of other elements cycle through complex chemical reactions that operate according to the unique conditions of pressure, temperature, and isolation that characterize this hidden world. The chemical composition of this hidden reservoir reads like an inventory of Earth's ancient geological and biological history, each dissolved element telling part of the story of how this system formed and evolved. Iron compounds, slowly dissolved from the surrounding bedrock over millions of years, give the water a metallic character that speaks of deep geological processes operating on timescales beyond human comprehension. Sulfur compounds, remnants of ancient biological process and geological interactions between water and rock, add their own distinctive signature to this primordial chemical soup. Organic carbon molecules, the molecular memory of life forms that thrived here when Antarctica enjoyed a much warmer climate, continue to fuel chemical reactions and biological processes in this dark world. The boundaries of this subglacial lake are not fixed and defined like those of surface water bodies we might be familiar with. Instead, they shift and change as the glacier above moves and settles, creating a dynamic environment where the volume and shape of the water body respond to the slow but constant motion of the overlying ice. The ice ceiling above us might be smooth in some places, polished by the circulation of water over thousands of years. In other areas, the pressure and movement of the glacier might have carved the ice into fantastic formations, underwater ice caves, channels, and chambers that create a complex three-dimensional environment far more intricate than early researchers imagined. Here and there throughout the system, the immense pressure might have forced water into smaller chambers and narrow channels, creating a network of connected pools and passages that extends far beyond the main lake basin. This network functions like an underground river system, but one made of liquid water flowing beneath and through solid ice rather than through soil and rock. The total extent of this subglacial water system remains unknown, as mapping such an environment presents extraordinary technical challenges, but researchers estimate it may include multiple connected water bodies spanning several square kilometers. The temperature gradients within this hidden world create their own mysterious dynamics that drive circulation patterns and chemical processes. While the average temperature hovers around -5 degrees Celsius, different areas of the lake system experience subtle variations due to geothermal heat rising from the bedrock below, pressure changes caused by the movement of ice above, and the slow circulation of water through the system. These temperature differences, though measured in just a few degrees, create currents and circulation patterns within the lake, a slow, ancient dance of water movement that has continued for millions of years, mixing chemicals and nutrients throughout the system. The pressure within this subglacial environment is immense yet remarkably stable, creating conditions unlike anything found in surface water bodies. The weight of the glacier above creates a constant downward force measured in hundreds of atmospheres. But this pressure is balanced by the incompressible nature of liquid water below. This creates a unique equilibrium, a state where the water exists in a sort of suspended animation, neither fully liquid in the sense we normally understand it, nor solid, but something in between that can only exist under these extraordinary conditions of pressure and chemistry. Within this hidden reservoir, time itself seems to move differently than in the surface world above. Changes occur on geological timescales, measured not in days, seasons, or years, but in millennia and millions of years. The slow circulation of water through the system, driven by minute temperature and pressure differences, might take centuries or even millennia to complete a single cycle. Chemical reactions proceed at rates determined not by heat or biological catalysts, but by the gradual availability of reactants and the persistent but gentle pressure of the environment. Even the life forms that inhabit this realm operate on timescales that dwarf human experience, with generations spanning years or decades rather than the days or weeks typical of surface organisms. The interaction between the water and surrounding rock and ice creates what could be described as a complex chemistry laboratory operating in slow motion over geological time. Iron minerals from the ancient bedrock slowly dissolve into the brine, through processes of chemical weathering that operate grain by grain, molecule by molecule. Organic compounds from ancient marine sediments break down and recombine through complex chemical pathways, creating new molecular species that exist nowhere else on Earth. These reactions produce the chemical precursors to the dramatic color changes that will occur when this water finally reaches the surface and encounters the oxygen-rich Antarctic atmosphere. The volume of this hidden reservoir has been a subject of scientific investigation and educated speculation, as determining the exact size and extent of a water body buried beneath hundreds of meters of ice presents extraordinary challenges. While the exact dimensions remain uncertain due to the technical difficulties of studying such an inaccessible environment, researchers using ground-penetrating radar and other geophysical techniques estimate that the lake system may contain millions of liters of this ancient brine. The water extends not just as a single large pool, but through a complex three-dimensional network of interconnected chambers, channels, and reservoirs carved into the bedrock and ice over millions of years. The hydrological connections within this subglacial system form a remarkably complex network that scientists are still working to map and understand completely. Water doesn't simply sit stagnant in this hidden world, but rather flows through an intricate maze of pathways that connect different parts of the system. Some routes lead deeper into the glacier system, connecting with other hidden water bodies that may exist elsewhere beneath the East Antarctic ice sheet. Others wind their way gradually toward the glacier's terminus, following fractures and weak points in the ice structure that will eventually allow some of this ancient water to see daylight for the first time in over 1 million years. The transformation that creates the haunting crimson cascade of blood falls begins with one of the universe's most abundant and fundamental elements, iron. Let us settle gently into the story of this ancient metal and its remarkable chemical journey from the hidden depths beneath Taylor Glacier to the stunning visual display that has captivated every visitor fortunate enough to witness this Antarctic wonder. The tale of iron at Blood Falls is ultimately a story about time, chemistry, and the patient processes that can create beauty from the most basic building blocks of matter. In the perpetual darkness of the subglacial reservoir, iron exists in a form that would surprise most of us who are accustomed to seeing this metal in its familiar surface manifestations. Here, dissolved in the ancient brine under conditions of low oxygen and high pressure, iron atoms float freely in their reduced state, known to chemists as ferrous iron or iron 2. In this chemical form, the iron is essentially invisible to the human eye. The water carrying these dissolved iron compounds appears clear and unremarkable, giving no hint of the spectacular transformation that awaits when this solution finally encounters the oxygen-rich environment of the Antarctic surface. The ultimate source of all this dissolved iron lies deep in the geological foundation we explored earlier, embedded in the bedrock and ancient marine sediments that form the floor of the subglacial lake system. The rocks beneath Taylor Glacier contain abundant iron-bearing minerals that formed under the reducing conditions of ancient marine environments millions of years ago. Picture this process of iron dissolution as an incredibly patient extraction, operating on timescales that dwarf human experience, where water gradually pulls iron atoms from solid rock formations, grain by grain, molecule by molecule, creating a solution that becomes increasingly rich in dissolved metals over geological time. The specific mechanisms of iron dissolution that occur in the subglacial environment are fascinating in their complexity and operate according to chemical principles quite different from the rapid rusting we might observe when iron meets oxygen and water in our everyday surface world. Unlike the familiar oxidation processes that create rust on metal objects exposed to air and moisture, the processes beneath the glacier occur in an environment largely devoid of free oxygen. Instead, iron is released through different chemical pathways that involve the breakdown of iron-bearing minerals like pyrite, magnetite, and various iron sulfide compounds embedded in the ancient marine sediments. These dissolution processes are mediated by the unique chemistry of the subglacial brine, which contains organic acids produced by microbial activity, sulfur compounds that can participate in complex chemical reactions, and other dissolved species that facilitate the gradual weathering of iron-bearing minerals. The low pH conditions created by these dissolved compounds help to keep iron in its reduced, soluble form, allowing it to accumulate in the water over millions of years. The result is a concentration of dissolved iron far exceeding what would be found in typical surface waters, creating the chemical preconditions for the dramatic visual display that occurs when this water finally reaches the surface. As the iron-rich brine begins its slow ascent through the glacier's internal pathways toward the surface, it encounters its first significant chemical challenge: the gradual introduction of oxygen into the system. The journey from the main subglacial reservoir to the surface outlet involves passage through fractures and channels within the glacier ice, and this ice contains tiny bubbles of ancient air that were trapped when the ice first formed hundreds or thousands of years ago. These microscopic air bubbles, compressed under the immense weight of the glacier, slowly release their oxygen into any water that comes into contact with them. The chemical reaction between dissolved ferrous iron and oxygen represents one of chemistry's most fundamental and visually dramatic transformations. This process, which scientists call oxidation, involves the transfer of electrons from iron atoms to oxygen molecules, fundamentally changing the chemical nature of the iron and creating entirely new compounds with different physical and optical properties. When ferrous iron encounters oxygen, it transforms from its reduced iron 2 state to the oxidized iron 3 state, and in the process creates various iron oxide and iron hydroxide compounds that are responsible for the spectacular coloration we observe. The visual transformation that results from this chemical reaction is nothing short of magical to witness. Scientists have confirmed that the stunning red coloration of blood falls results from the oxidation of iron compounds. As dissolved iron meets oxygen and undergoes rapid chemical transformation, the water emerges from the glacier clear or only slightly tinted, but quickly turns crimson as the oxidation reaction proceeds with surprising speed once the water reaches the oxygen-rich Antarctic atmosphere. Imagine watching invisible ink revealing its secret message. The clear water blossoms into brilliant shades of rust, orange, and deep red as billions of iron atoms undergo their chemical transformation simultaneously. The specific shades and intensity of color that develop depend on several interrelated factors, creating a natural palette that can range from bright orange to deep crimson to rich rust brown depending on conditions. The concentration of iron in the water directly influences the intensity of coloration. Higher concentrations of dissolved iron produce more dramatic and vivid colors, while lower concentrations result in more subtle tints and shades. The pH of the water affects which specific iron oxide compounds form during the oxidation process, and different iron compounds produce subtly different hues and color characteristics. The temperature of both the air and water influences the speed and completeness of the oxidation reactions, affecting how quickly the color change occurs and how intense the final coloration becomes. During the warmer Antarctic summer months, higher temperatures can accelerate the chemical reactions, potentially creating more intense coloration in a shorter period of time. Conversely, during the colder winter months, the reactions may proceed more slowly, creating more gradual color transitions and potentially different final color characteristics. The iron oxides that form during this oxidation process are chemically identical to the compounds responsible for common rust, but in the unique conditions of blood falls, they create something far more beautiful and scientifically interesting than the simple corrosion we typically associate with oxidized iron. The oxides form as extremely small particles suspended in the flowing water, creating what chemists call a colloidal solution. These microscopic particles interact with the light in complex ways, scattering and absorbing different wavelengths to produce the characteristic coloration that gives blood falls its dramatic appearance. These iron oxide particles are so small, measured in nanometers, that they remain suspended in the flowing water rather than settling out like heavier sediments might. This colloidal suspension maintains the vivid color throughout the waterfall's cascade and in the pools that form at the base of the falls. The particles continue to interact with light as they flow, creating shifting patterns of color and intensity that ensure no two photographs of blood falls ever look exactly the same. The chemistry becomes even more complex and interesting when we consider the other elements and compounds present in the ancient brine that participates in these reactions. Sulfur compounds, dissolved from the same geological sources as the iron, participate in their own chemical reactions when exposed to oxygen. Some of these sulfur-based reactions actually help to create and maintain the conditions that keep iron in its reduced, dissolved form while it remains in the oxygen-poor, subglacial environment. It's only when these sulfur compounds also encounter oxygen-rich conditions that the delicate chemical balance shifts, allowing the iron oxidation reactions to proceed rapidly. The precise timing and location of these chemical reactions create the distinctive visual pattern that characterizes blood falls and makes it such a compelling natural phenomenon. The water emerges from the glacier essentially clear or only slightly tinted, then undergoes its dramatic color transformation as it flows down the ice face and encounters increasing amounts of atmospheric oxygen. This creates a beautiful gradient effect where the water becomes progressively more colorful as it descends, with the most intense coloration typically occurring in the pools and streams at the base of the falls where the water has had the most time and opportunity to interact with oxygen-rich air. The iron chemistry of blood falls also involves chemical processes that occur on different time scales, adding layers of complexity to the overall phenomenon. The initial dramatic color change happens relatively rapidly, within minutes or even seconds of exposure to oxygen-rich air, but there are additional, slower reactions that continue to occur as the water flows and pools at the base of the falls. Over hours and days, additional iron compounds can form through continued oxidation and interaction with other dissolved species, creating the permanent reddish-brown staining that marks the glacier face and surrounding rocks, serving as a lasting record of millions of years of intermittent flow. In the absolute darkness beneath Taylor Glacier, where no photon of sunlight has penetrated for over 1 million years and temperatures hover far below the normal freezing point of water, life has not merely found a way to survive, it has created one of Earth's most remarkable ecosystems. Let us descend gently into this hidden microbial world, where some of our planet's most extraordinary organisms have been quietly living their fascinating lives in conditions that challenge our very definition of what makes an environment habitable for life. The discovery that Blood Falls harbors a thriving microbial community revolutionized scientific understanding of life's limits and possibilities. Chemical and microbial analyses revealed that this subglacial ecosystem contains autotrophic bacteria that have developed the remarkable ability to metabolize sulfate and ferric ions, essentially eating minerals and breathing chemicals that would be toxic to most life forms on Earth's surface. This represents what scientists recognize as one of the most unique biological communities on our planet, operating according to biological principles fundamentally different from the photosynthesis-based ecosystems that dominate the surface world. The microorganisms living in this subglacial environment face a constellation of challenges that seem almost insurmountable when viewed from the perspective of surface life. They exist in perpetual darkness, completely cut off from the sunlight that powers most of Earth's ecosystems through the fundamental process of photosynthesis. The water around them is not just cold, but maintains temperatures far below the point at which most life forms can maintain the basic cellular processes necessary for survival. The chemistry of their environment is extreme beyond most earthly comparisons, with concentrations of salt, metals, and other compounds that would prove immediately lethal to the vast majority of organisms that inhabit more hospitable environments. Yet somehow, these microscopic pioneers have not only adapted to these seemingly impossible conditions, they have transformed them into a thriving home where life flourishes in ways that continue to amaze and inspire scientists. Water samples collected from Blood Falls have revealed at least 17 different types of microorganisms, each representing a unique, evolutionary solution to the extraordinary challenges of life in this subglacial world. This hidden biodiversity was completely unexpected, demonstrating that even in Earth's most extreme environments, life finds ways to diversify and specialize in remarkable fashion. Each of these microbial species has evolved specialized strategies for extracting energy and nutrients from their harsh environment. Some have become experts at processing sulfur compounds, dissolved from the ancient geological formations, using complex biochemical pathways to extract energy from chemical reactions involving sulfur-based molecules. Others focus their metabolic efforts on iron compounds, manipulating the chemistry of iron ions through sophisticated cellular machinery that allows them to derive energy from reactions that would be impossible for surface organisms. Still others have developed ways to break down and utilize organic compounds that have been preserved in the ice for millions of years, essentially feeding on the molecular remains of ancient life forms. The dominant organism in this remarkable community has been identified through genetic analysis as closely related to Thiomicrospira arctica, a psychrophilic marine bacterium that represents one of nature's most accomplished cold-weather specialists. The bacterial 16S rRNA gene analysis revealed that this organism shares 99% genetic similarity with known psychrophilic marine autotrophic sulfur oxidizers, placing it within a family of bacteria specifically adapted to cold temperatures and capable of manufacturing their own nutrients without relying on organic food sources produced by other organisms. This remarkable bacterium serves as the foundation of the Blood Falls ecosystem, functioning as a primary producer in a world without sunlight. Through the process of chemosynthesis, it manufactures its own nutrients using the chemical energy available in the dissolved minerals and compounds of the subglacial brine. This ability to create organic compounds from inorganic raw materials makes it functionally equivalent to plants in surface ecosystems, but operating according to completely different principles that rely on chemistry rather than sunlight as the fundamental energy source. The metabolism of these subglacial organisms operates on principles that are fundamentally different from the photosynthesis that drives most surface ecosystems and that most of us learn about in basic biology classes. Instead of capturing energy from sunlight and converting carbon dioxide into organic compounds, these extremophiles obtain their energy through chemosynthesis, a process where they harness the chemical reactions between iron and sulfur compounds in the mineral-rich brine to produce the energy they need for growth, reproduction, and all the basic processes of life. Imagine these microscopic organisms as incredibly sophisticated chemical factories operating in slow motion in the frigid darkness of the subglacial environment. They carefully extract iron ions from the surrounding water, manipulate sulfur compounds that have been dissolved from ancient geological sources over millions of years, and orchestrate complex biochemical reactions that extract just enough energy to power their cellular processes. The efficiency required for survival in such an energy-poor environment has driven these organisms to become some of the most biochemically sophisticated life forms on our planet. The pace of life in this hidden ecosystem is almost incomprehensibly slow compared to what we observe in warmer, more energy-rich environments. These bacteria might undergo cellular division only once every few months or even years, making their community one of the slowest-growing ecosystems on Earth. This glacial pace of reproduction and growth reflects the fundamental energy constraints of their environment, where every chemical reaction must be carefully optimized to extract the maximum possible energy from the limited resources available. The adaptation strategies that these organisms have developed represent remarkable examples of evolutionary creativity operating under extreme selective pressure. Life in these extremely cold, low-nutrient environments has produced extremophilic microorganisms known as psychrophiles, organisms that have not just adapted to survive in cold conditions, but actually require cold temperatures to function properly. Their cellular machinery has been fundamentally redesigned to operate efficiently at temperatures that would quickly kill most other life forms. Their cell membranes have been extensively modified to remain flexible and functional at extremely low temperatures, incorporating specialized fatty acids and membrane proteins that prevent the cell membrane from becoming rigid and brittle in the cold. Normal cell membranes would freeze solid at the temperatures found in the blood falls ecosystem. But these specialized membranes maintain the fluid properties necessary for nutrients to pass in and waste products to pass out, allowing the cells to continue functioning even in conditions of extreme cold. The proteins produced by these organisms represent millions of years of evolutionary refinement for function at low temperatures. Most proteins would become denatured and non-functional at the temperatures found beneath Taylor Glacier, but these organisms produce specialized proteins that have been optimized through evolutionary processes to maintain their three-dimensional structure and catalytic activity even in extreme cold. Some of these proteins have been found to have unique structures and properties not found in any other organisms on Earth. The genetic machinery of these subglacial microorganisms represents millions of years of evolution in complete isolation from surface life forms. Cut off from genetic exchange with organisms in other environments, the microbial community beneath Taylor Glacier has evolved along its own unique evolutionary pathway, developing genetic solutions to environmental challenges that exist nowhere else on our planet. Some of their genes show adaptations that are completely novel, representing entirely new approaches to the fundamental challenges of life in extreme environments. Research has identified a diverse range of microorganisms thriving in this unique ecosystem, including various strains of bacteria and archaea that continue to flourish within the specific ecological framework of the subglacial habitat. The presence of archaea, ancient microorganisms that represent one of the three fundamental domains of life, alongside bacteria and eukaryotes, adds another layer of complexity and biological significance to this hidden ecosystem. These archaea possess their own unique biochemical pathways and may play crucial roles in cycling nutrients and maintaining the delicate chemical balance of the subglacial environment. The food web that operates in this dark world functions without the familiar foundation of plant photosynthesis that characterizes virtually all surface ecosystems. Instead, the chemosynthetic bacteria serve as primary producers, creating organic compounds from inorganic raw materials through their specialized metabolic pathways. These organic compounds then become available as food sources for other microorganisms in the community, creating a complex web of interdependencies that has remained stable for millions of years. Some bacterial species in the community specialize in breaking down complex organic molecules that have been preserved from the ancient marine environment, essentially recycling nutrients that originated from life forms that existed millions of years ago. Others focus on processing the waste products and dead cellular material produced by their microbial neighbors, ensuring that nothing of nutritional value is lost in this resource-constrained environment. This creates an intricate web of chemical interdependencies where each species plays a specific role in maintaining the overall health and stability of the community. The reproduction and growth strategies employed by these organisms reflect the extreme constraints and challenges of their environment in fascinating ways. With energy sources severely limited and chemical processes proceeding slowly in the extreme cold, these microorganisms have evolved to be incredibly efficient in their use of available resources. They waste virtually nothing, developing sophisticated cellular machinery to recycle every available nutrient and chemical compound. Their reproduction rates are so slow that each new generation represents a significant investment of the community's limited energy and material resources. The journey from mysterious discovery to scientific understanding required decades of carefully planned expeditions into one of Earth's most challenging and remote environments. Let us follow the brave researchers who ventured repeatedly into the Antarctic wilderness, carrying with them sophisticated scientific equipment, unwavering determination, and the curiosity needed to unlock the secrets of Blood Falls. These expeditions represent some of the most challenging scientific fieldwork ever undertaken, combining cutting-edge research techniques with the physical and logistical demands of working in one of the planet's most unforgiving environments. The early scientific expeditions to Blood Falls were exercises in both scientific curiosity and human endurance, requiring researchers to push the boundaries of what was technically possible in Antarctic field research. Picture the enormous logistical challenges facing scientists who wished to study this remote phenomenon in detail, located hundreds of kilometers from the nearest permanent research station in a region where weather conditions could turn life-threatening within minutes, and where every piece of equipment had to function reliably in conditions far outside normal operating parameters. Dr. Jill McCookie from the University of Tennessee emerged as one of the pioneering researchers dedicated to solving the Blood Falls mystery through rigorous scientific investigation. Her expeditions beginning in the early 2000s marked the beginning of serious, systematic scientific investigation into the phenomenon's true nature, moving beyond speculation and theory toward the kind of detailed sampling and analysis that could provide definitive answers. McCookie and her interdisciplinary research teams faced the unique challenges of conducting delicate microbiological and chemical sampling in an environment where temperatures could drop to dangerous levels within minutes, and where every piece of equipment had to be carefully protected from the harsh Antarctic conditions that could destroy sensitive instruments. The logistics of reaching Blood Falls began long before the actual fieldwork, requiring months of planning and coordination with multiple agencies and institutions. Research teams had to coordinate with the United States Antarctic Program, securing the necessary permits and clearances, arranging helicopter transportation from McMurdo Station, and carefully planning supply chains that could support weeks of intensive field research in one of the world's most remote and inaccessible locations. Every tool, every sample container, every piece of protective equipment, and every backup system had to be considered absolutely essential, as there would be no opportunity to replace forgotten items or repair failed equipment once the expedition was underway in the field. The helicopter flights from McMurdo Station to Taylor Valley provided research teams with their first aerial perspective of the phenomenon they had come thousands of miles to study. Flying over the stark, otherworldly landscape of the McMurdo Dry Valleys, team members could observe Blood Falls as a distinctive dark stain against the brilliant white glacier, its reddish coloration clearly visible even from altitude against the pristine Antarctic landscape. These flights also revealed the broader geological context of the phenomenon, showing researchers how the falls fit into the complex system of glaciers, valleys, and exposed rock formations that characterize this unique region of Antarctica. Establishing a research camp near Blood Falls required careful consideration of both immediate scientific needs and critical safety requirements. The research teams had to position themselves close enough to the falls to conduct detailed sampling and continuous measurements, yet maintain sufficient distance to avoid the potential hazards associated with working near an active glacier where ice movement, sudden weather changes, and hidden crevasses could pose serious dangers. The wind patterns in Taylor Valley are notoriously unpredictable and can be fierce, with katabatic winds capable of creating whiteout conditions within minutes and generating wind chill factors that can cause frostbite in exposed skin within seconds. Every piece of scientific equipment and camping gear had to be carefully secured against these sudden and dramatic weather changes that characterize the Antarctic environment. Tents had to be anchored with specialized stakes designed for frozen ground. Equipment had to be stored in protective cases that could withstand extreme temperature fluctuations, and backup systems had to be in place for every critical piece of research equipment. The research teams learned to work efficiently during favorable weather windows, sometimes conducting intensive sampling sessions that lasted for many hours when conditions were good, then waiting out storms in heated shelters while processing samples and planning the next phase of their investigation. The actual sampling of Blood Falls water presented unique technical challenges that tested both the researchers' scientific ingenuity and their physical endurance in the harsh environment. The water emerging from the glacier was not just cold. It existed at temperatures well below the normal freezing point and remained liquid only due to its extraordinary salt content and the pressure conditions within the glacier. Standard water collection bottles and sampling equipment would freeze solid within minutes of exposure to the Antarctic air, making it necessary to develop specialized collection techniques and containers that could preserve samples in their liquid state long enough for transport back to heated laboratory facilities. Mikuki's team developed innovative methods for collecting and preserving the unique chemistry and biology of Blood Falls water, drawing on techniques from multiple scientific disciplines. They employed insulated sampling containers with specialized thermal properties, used heated collection systems powered by portable generators to prevent samples from freezing during the collection process, and developed protocols for rapid sample processing that could preserve both chemical and biological characteristics. Each sample had to be meticulously labeled and documented, recording not just what was collected, but also the exact environmental conditions under which the collection took place: air temperature, wind speed, time of day, flow rate of the falls, and detailed visual characteristics of the phenomenon at the moment of sampling. The microbial sampling required even more specialized techniques and equipment, as researchers understood that if microorganisms were present in the blood falls water, they would be adapted to very specific temperature, pressure, and chemical conditions that would have to be maintained throughout the collection, transport, and storage process. Preserving these potential organisms for laboratory analysis meant maintaining sample temperatures and chemical conditions as close as possible to their natural state throughout the entire chain of custody from collection site to laboratory analysis. This required portable refrigeration systems, specialized sample containers, and backup power systems that could function reliably in the extreme conditions of the Antarctic field environment. Documentation of the fieldwork involved extensive photography and video recording to capture both the visual characteristics of the phenomenon and the scientific methods being employed, but even these seemingly straightforward tasks presented significant challenges in the Antarctic environment. Camera batteries died quickly in the extreme cold, requiring researchers to keep spare batteries warm inside their insulated clothing. Condensation could form on camera lenses when equipment was moved between different temperature zones, and the brilliant Antarctic sunlight reflecting off snow and ice required careful exposure settings and specialized filters to capture accurate images of the fall's true coloration and flow characteristics. The research teams also conducted extensive measurements of the fall's flow rates, temperature profiles at various points along the flow path, and detailed chemical characteristics at the source and at multiple points downstream. This quantitative work required deploying sensitive scientific instruments directly in the path of the flowing, freezing water, often meaning that researchers had to work with wet, rapidly freezing hands while handling delicate electronic equipment and precision instruments. The dedication and persistence required to gather this detailed data under such challenging conditions exemplified the commitment of Antarctic researchers who understand that some fundamental scientific questions can only be answered through direct field investigation under extreme conditions. Weather conditions in the McMurdo Dry Valleys could transform the entire character of an expedition within hours, requiring research teams to remain flexible and prepared for rapid changes in their research plans. Clear, calm days perfect for detailed sampling and extended fieldwork could suddenly give way to fierce windstorms with sustained winds over 100 kilometers per hour that made any outdoor work extremely dangerous. Research teams learned to take full advantage of favorable weather windows, sometimes working extended shifts during good conditions and then weathering severe storms in their heated shelters, while processing samples, analyzing preliminary data, and planning the next phase of their investigation. The international collaborative aspect of Antarctic research meant that blood falls attracted scientific attention from research institutions around the world, creating opportunities for knowledge sharing and technical collaboration. Teams from multiple countries and research institutions brought different areas of expertise and different scientific perspectives to the investigation. Glaciologists worked alongside microbiologists, geochemists collaborated with climatologists, and atmospheric scientists partnered with molecular biologists. Each expedition built systematically upon the discoveries and refined techniques developed by previous research teams, creating a growing body of knowledge and increasingly sophisticated understanding of this remarkable phenomenon. Transportation of precious samples from the remote field site back to sophisticated laboratory facilities around the world required careful coordination with Antarctic logistics operations and strict adherence to protocols designed to preserve sample integrity. Samples had to be kept at precisely controlled temperatures during helicopter transport back to McMurdo Station, then maintained at appropriate temperatures during the long flights to New Zealand and ultimately to specialized research laboratories in the United States and other countries. The chain of custody for these irreplaceable samples was absolutely critical, as some planned experiments required samples that had never been exposed to temperatures above certain thresholds or had never been contaminated with surface microorganisms. The true secrets of blood falls could only be unlocked once the precious samples collected from the Antarctic wilderness reached sophisticated laboratory facilities around the world, where advanced analytical instruments could reveal the extraordinary chemical and biological truths hidden within each carefully preserved vial. Let us follow this scientific detective story as it unfolds in temperature-controlled research facilities far from the frozen continent, where cutting-edge technology and methodical scientific investigation would finally reveal the remarkable truth about this crimson mystery that had puzzled observers for over a century. The samples arriving at Dr. McCookie's laboratory at the University of Tennessee represented far more than just Antarctic water. They carried within their molecular structure the chemical and biological signatures of an ancient world that had been sealed away from surface processes for millions of years. The first critical priority was preserving the absolute integrity of these irreplaceable samples while subjecting them to the detailed analysis that would unlock their closely guarded secrets. This delicate balance required maintaining strict temperature controls throughout all handling procedures, working quickly to prevent any changes in the delicate chemistry that had been preserved for geological ages beneath the glacier, and using analytical techniques that could extract maximum information while consuming minimum amounts of the precious sample material. The initial chemical analysis immediately confirmed that Blood Falls water was unlike anything previously studied from Antarctic sources or from any other natural water body that scientists had encountered. The salinity levels were extraordinary, measuring nearly 3 times that of typical seawater, creating a brine solution so concentrated that it explained how the water could remain liquid at temperatures well below the normal freezing point of pure water. But it was the specific composition of dissolved minerals and the precise ratios of different chemical species that truly set this water apart from any other natural water system known to science. The iron concentrations in the samples were absolutely staggering, far exceeding what researchers had expected, even given the obvious iron-related coloration visible in the phenomenon itself. Advanced spectroscopic analysis revealed not just extremely high levels of total iron content, but a highly specific distribution of iron compounds that told a detailed story of complex chemical processes that had been occurring in the isolated subglacial environment over geological time. Most significantly, the iron in freshly collected samples existed primarily in its chemically reduced form, ferrous iron, which could only remain stable in the oxygen-poor environment of the subglacial lake system. When laboratory researchers deliberately exposed fresh samples to oxygen-rich air under carefully controlled experimental conditions, they could recreate and study in detail the dramatic color transformation that occurs naturally when the water emerges from the glacier and encounters the Antarctic atmosphere. Clear samples turned brilliant red within minutes under controlled laboratory conditions, confirming definitively that the fall's spectacular coloration resulted from the rapid oxidation of dissolved iron compounds, rather than from biological pigments, as had been theorized for decades. This elegantly simple but scientifically rigorous experiment proved once and for all that blood falls was fundamentally a chemical phenomenon rather than a biological one. The isotopic analysis of the water samples provided perhaps the most startling and scientifically significant revelation about the age and origin of the blood falls system. By examining the precise ratios of different isotopes of oxygen and hydrogen in the water molecules themselves, researchers could determine with remarkable accuracy how long the water had been completely isolated from the atmosphere and surface processes. The isotopic signatures indicated that the water had been trapped beneath the glacier for an estimated 1.5 to 2 million years, making it some of the oldest isolated liquid water ever discovered anywhere on Earth's surface. This extraordinary age meant that the water samples represented a direct, uncontaminated connection to Antarctica's ancient climate history, preserving chemical information about environmental conditions that existed long before modern ice ages and climate cycles. The chemical composition preserved in these samples reflected atmospheric and oceanic conditions that existed when the glacier first advanced far enough to seal off the subglacial environment permanently. In essence, each carefully labeled sample vial contained a liquid time capsule, preserving detailed information about Earth's climate, atmospheric composition, and ocean chemistry from an era when our planet was experiencing fundamentally different environmental conditions. The microbiological analysis of the samples proved to be even more revolutionary and paradigm-shifting than the remarkable chemistry results. When researchers first attempted to culture microorganisms from the blood falls water using standard laboratory techniques that had been successful with other extremophile organisms, they encountered unexpected and frustrating challenges. Standard laboratory protocols for growing microorganisms failed consistently because the controlled laboratory conditions were too warm, too rich in oxygen, and too nutritionally abundant compared to the extreme environment from which these specialized organisms had originated. Dr. Mikuki and her colleagues realized they would have to develop entirely new laboratory protocols and techniques specifically designed to successfully study the blood falls microorganisms in ways that respected their unique evolutionary adaptations. They created specialized culture media that precisely mimicked the high salt, low temperature, low oxygen conditions of the subglacial environment, working in specialized cold rooms and using custom-built incubators that could maintain the frigid temperatures these organisms required for basic survival and growth. Even with these careful adaptations to replicate their natural environment, the microorganisms grew incredibly slowly under laboratory conditions, requiring weeks or months to produce visible colonies that other bacteria might develop in just hours or days. The genetic analysis of these microorganisms revealed a hidden ecosystem of remarkable diversity and sophisticated biochemical capabilities that had never been observed in any other environment. Using advanced DNA sequencing techniques and comparative genomic analysis, researchers identified at least 17 different types of microorganisms living in the blood falls water, each representing a unique evolutionary lineage with its own specialized adaptations. Each species showed distinctive genetic adaptations to the extreme conditions of their subglacial home, including specialized proteins designed to function efficiently at very low temperatures and unique metabolic pathways specifically adapted to extract energy from the unusual chemical environment of the ancient brine. The dominant organism in this microbial community was identified through detailed genetic analysis as closely related to Thiomicrospira arctica, a psychrophilic bacterium known for its remarkable ability to derive energy from sulfur compounds rather than from organic matter or sunlight. This discovery was particularly significant because it confirmed that the blood falls ecosystem was based on chemosynthesis rather than the photosynthesis that drives virtually all surface ecosystems. These bacteria were essentially eating chemicals dissolved from the surrounding rock formations, converting inorganic mineral compounds into the organic molecules needed for growth, reproduction, and all the fundamental processes of life. Further analysis revealed the presence of archaea in the samples, ancient microorganisms that represent one of the three fundamental domains of life, alongside bacteria and eukaryotes. These archaea showed genetic characteristics and biochemical capabilities that had never been observed before in any other organism, suggesting that the millions of years of isolated evolution occurring beneath Taylor Glacier had produced completely unique evolutionary solutions to the challenges of life in extreme environments. Some of their genetic sequences were so distinctive that they represented entirely new branches on the tree of life. The metabolic analysis of these microorganisms provided detailed insights into the complex biogeochemical cycles operating in the subglacial environment, revealing that this was not simply a collection of organisms surviving in isolation, but rather a sophisticated ecosystem with intricate interactions between different species. The bacteria were not passive inhabitants of the unusual brine. They were actively modifying its chemistry through their metabolic processes, consuming sulfur compounds, processing iron ions, and producing organic compounds that served as nutrients for other members of the microbial community. This created a completely self-sustaining ecosystem that had been operating independently from surface processes for millions of years. Laboratory experiments designed to understand the microorganism's role in maintaining the chemical conditions necessary for the blood falls phenomenon revealed sophisticated interactions between biological and chemical processes. The bacteria played a crucial role in maintaining the reduced iron concentrations in the subglacial environment through their specialized metabolic activities, essentially creating and maintaining the chemical preconditions necessary for the dramatic color change that occurs when the water finally reaches the surface and encounters oxygen-rich air. The preservation of organic compounds in the samples opened an entirely new window into the ancient history of the blood falls system and the biological processes that had been occurring in this isolated environment. Advanced mass spectrometry analysis revealed the presence of complex organic molecules that had been preserved in the cold, oxygen-free environment for millions of years without significant degradation. Some of these compounds appeared to be molecular remnants of ancient marine life that had lived in the region before it was sealed beneath the advancing glacier, while others were clearly the products of ongoing microbial metabolism occurring in the current subglacial environment. Today, as we settle into our final exploration of the scientific understanding surrounding blood falls, we find ourselves with a remarkably complete picture of one of Antarctica's most extraordinary phenomena. The Crimson Cascade that once mystified early explorers and spawned decades of speculation now stands revealed as a profound window into deep geological time, extreme biological adaptation, and the hidden processes that continue to shape our planet in ways we are only beginning to understand. Let us gather together all the threads of this remarkable scientific story and appreciate how modern research has transformed mystery into comprehensive understanding. The current scientific consensus presents blood falls as a unique convergence point where geology, chemistry, and biology intersect and operate together on timescales that dwarf human experience and challenge our normal understanding of how ecosystems function. We now understand with considerable confidence that this phenomenon represents the surface expression of a vast subglacial ecosystem that has been evolving in complete isolation for over 1.5 million years. This ecosystem, permanently sealed beneath Taylor Glacier, has created its own distinctive chemical environment and biological community, developing and maintaining itself completely independently from the surface world above and all the climatic and environmental changes that have transformed Antarctica over geological time. The modern understanding of the fall's complex chemistry centers on the remarkable preservation of ancient seawater beneath the glacier, concentrated and chemically modified over millions of years of isolation. This brine solution, with a salinity nearly 3 times that of modern oceans, contains dissolved iron compounds that have been accumulating slowly but steadily for millions of years through the gradual weathering of iron-rich bedrock and ancient marine sediments. The iron exists primarily in its chemically reduced state in the oxygen-poor subglacial environment, remaining essentially invisible until the dramatic moment when it contacts the Antarctic atmosphere and undergoes rapid oxidation to create the spectacular red coloration that defines blood falls. Recent research utilizing advanced geophysical techniques has significantly refined our understanding of the complex pathway that brings this ancient water to the surface, where it can be observed and studied. Ground-penetrating radar studies have successfully mapped portions of the subglacial water system, revealing a far more extensive and complex network of channels and reservoirs than early researchers imagined possible. The water's journey from the main subglacial lake system to the surface outlet involves an intricate route through fractures, channels, and cavities within the glacier, with flow rates and intensity varying based on seasonal temperature changes, internal glacier dynamics, and pressure variations within the subglacial system. The microbial ecosystem discovered in Blood Falls water has fundamentally revolutionized scientific understanding of life's possibilities in extreme environments and expanded our conception of where life might exist throughout the universe. The community of at least 17 different microorganisms, dominated by chemosynthetic bacteria that derive their energy from sulfur and iron compounds rather than sunlight, represents one of Earth's most isolated and unique biological systems. These organisms have been evolving independently for millions of years, completely cut off from genetic exchange with surface life forms, developing extraordinary genetic adaptations that allow them to thrive in conditions of perpetual darkness, extreme cold, and high chemical stress that would quickly kill most other known life forms. Modern molecular biology techniques and advanced genetic sequencing have provided unprecedented insights into the genetic makeup and evolutionary history of these remarkable extremophiles. Comparative genomic analysis has revealed that many of the Blood Falls microorganisms possess genes and genetic sequences found nowhere else on Earth, representing completely unique evolutionary solutions to environmental challenges. Their proteins have been extensively modified through millions of years of natural selection to remain functional at temperatures that would denature the proteins of virtually all other life forms, and their metabolic pathways have been optimized for the slow-paced chemistry of their frigid, nutrient-limited environment. The crucial role of these microorganisms in maintaining the chemical conditions that make the Blood Falls phenomenon possible has become much clearer through recent research combining laboratory studies with field observations. The bacteria actively participate in complex biogeochemical cycles that preserve iron in its chemically reduced form within the subglacial environment, essentially creating and maintaining the chemical preconditions necessary for the spectacular surface display. Their specialized metabolic activities help maintain the unique chemistry of the subglacial brine, making them not just passive inhabitants of this extreme environment, but active participants in creating and sustaining the phenomenon we observe. Current research efforts have increasingly focused on understanding the broader implications of the blood falls ecosystem for astrobiology and the scientific search for life beyond Earth. The environmental conditions beneath Taylor Glacier, characterized by perpetual darkness, extreme cold, high chemical stress, and complete isolation from surface processes, are remarkably similar to environments that planetary scientists believe might exist beneath the ice shells of Jupiter's moon Europa or Saturn's moon Enceladus. The discovery of not just surviving but thriving life in such conditions has dramatically expanded our concept of potentially habitable environments and has directly informed the design of future space missions specifically designed to search for signs of extraterrestrial life. The technological advances that made the modern understanding of blood falls possible represent a remarkable convergence of multiple scientific disciplines and cutting-edge analytical techniques. Revolutionary advances in molecular biology enabled the identification, genetic analysis, and detailed study of extremophile microorganisms that cannot be studied using traditional techniques. Major improvements in analytical chemistry allowed for precise determination of the water's complex chemical composition and isotopic signatures that revealed its ancient age. Significant developments in remote sensing technology, including specialized ground-penetrating radar systems and advanced satellite imagery, provided new tools for mapping the subglacial environment that feeds the falls and understanding its connection to broader Antarctic ice sheet dynamics. The isotopic analysis that revealed the extraordinary ancient age of the Blood Falls water has also provided valuable insights into Antarctica's complex climate history over millions of years. The water trapped beneath Taylor Glacier preserves detailed chemical signatures from a time when Antarctica was experiencing very different climate conditions, offering researchers a unique and irreplaceable window into the continent's environmental past. This information contributes significantly to broader scientific understanding of how Antarctic ice sheets have responded to climate change over geological time and provides important context for understanding current and future climate change impacts. Recent sophisticated modeling studies have significantly improved scientific understanding of how the blood fall system maintains its remarkable stability over such extraordinarily long timescales. Advanced computer simulations of the subglacial hydrology demonstrate how the system can persist and maintain its essential characteristics despite gradual changes in glacier thickness, regional temperature patterns, and other environmental conditions that have varied over geological time. These models help explain how this unique ecosystem has survived through multiple glacial and interglacial periods that have dramatically altered Antarctica's surface conditions while leaving the subglacial environment relatively undisturbed. The discovery and analysis of complex organic compounds preserved in the Blood Falls water has opened entirely new avenues for understanding how sophisticated organic molecules can be preserved over geological time in extreme environments. Some of these compounds appear to be molecular remnants of ancient marine life that existed in the region millions of years ago, while others are clearly the products of ongoing microbial metabolism occurring in the current subglacial ecosystem. This research has important implications for understanding how organic molecules and potentially even biosignatures might be preserved in similar extreme environments elsewhere in the solar system. Modern continuous monitoring of blood falls has revealed subtle but important patterns in its flow characteristics and visual appearance that reflect both seasonal environmental variations and longer-term changes in the glacier system that feeds it. Automated weather stations and remote sensing equipment now provide continuous data streams on temperature, wind patterns, humidity, and other environmental factors that influence the fall's behavior over daily, seasonal, and annual cycles. This comprehensive monitoring has demonstrated that while the phenomenon responds measurably to environmental changes, it does so within the context of a remarkably stable system that has maintained its essential characteristics for millions of years. The educational and public outreach value of blood falls has grown tremendously alongside scientific understanding of the phenomenon and its broader implications. The visually striking appearance of the falls, combined with the remarkable scientific story revealed through decades of careful investigation, has made it an exceptionally powerful example of how modern interdisciplinary science can solve complex natural mysteries. The complete story of blood falls effectively demonstrates the critical importance of interdisciplinary research approaches, international scientific cooperation, and the technological advances that enable exploration and understanding of Earth's most extreme and challenging environments. Current conservation efforts focus intensively on protecting the blood falls site and its unique ecosystem from potential human impacts, as interest in Antarctic tourism and research continues to grow. As both scientific interest and public awareness of this remarkable phenomenon increase, researchers and environmental policymakers work collaboratively to ensure that this irreplaceable natural laboratory remains completely undisturbed for future scientific study and discovery. The site's formal designation as part of the Antarctic Specially Protected Area System helps preserve both the physical phenomenon itself and the crucial scientific opportunities it represents for ongoing research. As we reach the end of our gentle journey through the remarkable story of blood falls, let us rest here together in quiet contemplation of this extraordinary tale that bridges millions of years of Earth's history, connects multiple scientific disciplines, and reminds us of the profound mysteries that still lie hidden within our own planet, waiting patiently for curious minds to discover their secrets. The story of blood falls invites us to reflect deeply on the fundamental nature of scientific discovery itself, and the patient, methodical process by which human understanding gradually expands. For over a century, this crimson cascade captured human imagination with its striking beauty and seeming impossibility. Against the endless white expanses of Antarctica's pristine landscape, here was something that seemed almost alive, almost magical, a vivid splash of color in a world defined by ice, stone, and the austere beauty of extreme environments. What strikes us most profoundly about the blood falls story is how it demonstrates the patience and persistence required for true scientific understanding to emerge. The mystery persisted through decades of speculation, partial explanations, and gradual advancement of scientific knowledge and technology. The red algae theory served science well for many years, providing a logical framework that helped researchers think productively about life in extreme environments and develop testable hypotheses. Yet this explanation, while reasonable given the scientific knowledge and analytical capabilities of its time, ultimately gave way to a truth far more extraordinary and scientifically significant than anyone had dared to imagine. The revelation that blood falls represents a window into a hidden world that has existed in complete isolation for over 1 million years challenges us to consider the vast timescales on which our planet's most important processes operate. While human civilizations rise and fall over centuries and millennia, the ecosystem beneath Taylor Glacier has maintained its quiet existence through countless cycles of climate change, geological upheaval, and evolutionary transformation occurring in the world above. In the perpetual darkness beneath the ice, time moves according to different rhythms, measured not in human generations, but in geological ages that span millions of years. The microorganisms thriving in this subglacial realm offer us profound and inspiring lessons about life's remarkable creativity, adaptability, and persistence in the face of seemingly insurmountable challenges. These microscopic beings, invisible to the naked eye, represent some of the most accomplished survival artists on our planet. They have transformed what would seem like impossible obstacles, perpetual darkness, bitter cold, toxic chemistry, and complete isolation, into the foundation for a thriving, sophisticated ecosystem that has persisted for geological ages. There is something deeply moving and humbling about the fact that these organisms have been quietly living their lives, generation after generation, in complete isolation from the surface world and all its changes. They existed through ice ages that transformed entire continents, through the evolution of complex life forms, through the eventual emergence of human consciousness that would one day develop the scientific tools and curiosity needed to discover their hidden realm. Yet through all these surface changes, they persisted, adapted, and evolved, creating their own unique solutions to the fundamental challenges of existence in one of Earth's most extreme environments. The chemical transformation that creates bloodfall's dramatic coloration speaks to the fundamental forces that shape our world and create natural beauty through the most basic processes of chemistry and physics. The conversion of invisible dissolved iron into brilliant crimson oxides represents chemistry operating as art, physics performing as theater. Each time ancient water emerges from its glacial prison and meets the Antarctic air, we witness a reaction that has been occurring for millions of years. The same elemental dance between iron and oxygen that has been painting landscapes and creating geological formations since the early history of our planet. The scientific journey that led to understanding blood falls reminds us powerfully of the importance of human curiosity, international collaboration, and the sharing of knowledge across traditional boundaries. Researchers from multiple disciplines and many different countries worked together over decades, sharing insights, techniques, and discoveries across the artificial boundaries that often separate scientific fields. Geologists collaborated with microbiologists, chemists worked alongside glaciologists, and Antarctic field researchers partnered with laboratory scientists around the world. This collaborative spirit demonstrates how the most profound and meaningful discoveries often emerge when we combine different perspectives, expertise, and ways of understanding the natural world. The technological advances that made the blood falls discoveries possible also reflect humanity's remarkable ingenuity in developing tools and techniques to explore the unknown and answer fundamental questions about our world. From sophisticated analytical instruments that revealed the water's ancient chemistry to genetic sequencing techniques that identified the hidden microbial ecosystem, each technological breakthrough opened new windows of understanding. These same tools, approaches, and scientific methods now guide our search for life beyond Earth, as we apply the lessons learned from blood falls to exploration of potentially habitable environments on distant moons and planets throughout our solar system. The preservation of blood falls as a natural laboratory and protected scientific site reminds us of our responsibility as careful stewards of Earth's remarkable phenomena and unique ecosystems. This extraordinary ecosystem, which remained undisturbed for millions of years, now exists in a world where human activities can potentially reach even the most remote corners of our planet. The thoughtful protection of sites like blood falls ensures that future generations of scientists will have opportunities to continue learning from these natural wonders, and that new technologies and analytical techniques yet to be invented might reveal even deeper secrets hidden within their ancient waters. As we consider the broader implications and deeper meaning of the Blood Falls story, we find ourselves contemplating questions that reach far beyond this single Antarctic phenomenon. If life can thrive in such extreme conditions right here on our own planet, what extraordinary possibilities might exist for life elsewhere in the vast universe? The organisms beneath Taylor Glacier have demonstrated conclusively that life can persist and flourish in conditions remarkably similar to those that might exist beneath the ice shells of Europa, Enceladus, and other potentially habitable worlds throughout our solar system and beyond. The timescales involved in the Blood Falls story, millions of years of isolation, ancient water preserved like liquid time itself, chemical processes operating with geological patience and persistence, invite us to consider our own place in the vast sweep of planetary history. Our human experience, measured in decades and centuries, represents just the briefest moment in the story of this ancient ecosystem. Yet through science, imagination, and careful investigation, we can reach across those immense expanses of time and connect meaningfully with processes and organisms that predate our species by millions of years. The beauty of Blood Falls, both visual and scientific, serves as a powerful reminder that our world continues to hold countless mysteries and wonders waiting to be discovered and understood. Even in an age when we can map our planet from space, peer into the depths of distant galaxies, and analyze the genetic code of life itself, remarkable secrets remain hidden beneath our feet, in the deep ocean and in the frozen corners of our continents. The story of Blood Falls encourages us to remain curious, to question our assumptions, and to remember that the most extraordinary discoveries often await us in places we least expect to find them. As you settle into peaceful sleep tonight, perhaps you can carry with you the inspiring image of that crimson water flowing steadily from its ancient source, bridging vast spans of time with each drop that catches the pale Antarctic light. In its patient flow, we see the remarkable persistence of life, the fundamental power of chemistry, the slow patience of geology, and the endless creativity of the natural world. Blood Falls reminds us that our planet remains full of wonder, mystery, and stories yet to be told by future generations of curious scientists and explorers. May your dreams tonight be filled with the quiet marvels of our natural world, the joy of discovery and understanding, and the peaceful recognition that we share our remarkable planet with extraordinary forms of life that continue to surprise and inspire us. Sleep well, knowing that somewhere in the darkness beneath Antarctic ice, ancient waters continue their patient journey, carrying precious secrets from deep time toward the light of human understanding. Rest peacefully and dream of crimson waterfalls, hidden worlds, and the countless mysteries that surround us, waiting patiently for curious minds to discover their secrets and share their stories with the world. Good night!